Hi. It's my distinct privilege and pleasure to be able to present to you today. And also, I have to say, it's a real privilege to co-direct this institute with all of the people that you're hearing from today, because in addition to being close colleagues and friends, all of them have been really amazing mentors to me the whole time I've been at UCSF, so I will not forget. Our module is focused almost exclusively on this one particular question. How can we determine if our therapies are having a meaningful impact on the latent HIV reservoir? So you've heard throughout the course of the day that this population of latently infected cells is what stands between us and a cure for HIV infection. But this concept of the latent reservoir is still a bit of a nebulous concept. And so we really need to devise accurate and sensitive tools to gauge the size of the reservoir so that we can figure out if any of the tools that we're using in the clinic are making a meaningful difference and if they're getting us closer to a cure. And I'd say the tools that we're developing within our component of the Institute will drive the work in all of the other modules. So in terms of what Warner presented to you today, the tools that we're developing will help us to gauge whether the candidate latency reversal strategies that are being developed in Warner's module are doing what they need to destroy latently infected cells. Then our tools will definitely help to drive the work that Dr. McCune just presented to you. A lot of these tools will be used to interrogate latent reservoirs within tissue sites themselves. And as you'll hear after my talk, Steve Deeks is going to talk about where the rubber hits the road, so essentially the clinical experiments that we'll be trying. The tools that we develop here, the goal is to develop scalable strategies to measure the latent reservoir so that we can determine if any of the interventions that we're trying in the clinic are legitimately reducing the size of the latent reservoir. And ultimately, we're hoping to use these tools to determine if we can safely stop antiretroviral therapy in an individual and expect to see a long remission period and a period of viral control in the absence of any continuous antiretroviral therapy. So the first thing you have to do to embark on an adventure like this is assemble a team of just legendary world-class scientists. And I have the privilege of working with a very diverse and brilliant panel of scientists, including clinicians, hardcore basic scientists, and computational scientists as well, from both the academic sector as well as the private sector in and around the Bay Area. So this is definitely a very overused metaphor, but this is definitely the time and place for it. The first challenge that faces the idea of measuring the latent reservoir is that HIV persists at low levels during antiretroviral therapy. So this is definitely a classic case of the needle in the haystack problem. So just to put some numbers to this in the next few slides, HIV-infected cells are extremely rare in a typical antiretroviral therapy suppressed individual. So let's try to embrace how rare these cells actually are. So if we take a sample of blood and we focus specifically on white blood cells, most of the cells within a sample of blood are irrelevant to HIV infection. So most of those cells are not susceptible to HIV infection and do not carry HIV. And there was a really nice conversation about this earlier, but CD4 T cells are the principal target for HIV infection in peripheral blood. Not the exclusive target, but definitely the principal target for HIV infection. So already we're getting down to about 10% of the white blood cells that could potentially serve as targets for HIV infection. Now to get down even further, as few as 10 out of 1 million CD4 T cells actually harbor HIV DNA in an HIV-infected individual on antiretroviral therapy. So these infected cells are few and far between, and the body is a very large and complex habitat. So we desperately need to develop tools that are extremely sensitive to identify these latently infected cells. So here's an additional challenge. The tools that we actually use to measure HIV DNA in the laboratory can only consider a very small number of cells at any given time. So not only are these cells really rare, we can only focus on a handful of cells in the laboratory to evaluate the presence of HIV. So it is very easy to imagine that we will actually fail to detect latently infected cells in a patient even if they are present. And I'd like to remind everybody that this is not a simple academic concern. This is a very real-world, important concern. So there have been multiple cases now where an individual was suspected of being cured. And when I say cure, I mean really have undergone complete viral eradication where there's no remaining traces of HIV RNA and DNA. So one very famous example is the case of the Mississippi baby, where at least at a few different time points, there was no evidence of any remaining HIV DNA or RNA in the Mississippi baby. But sure enough, and it took some time, but eventually when we stopped antiretroviral therapy, within a couple of years, the virus came back. So the critical lesson there is the virus was lurking somewhere, but the tools that we were using to interrogate this population of latently infected cells were not sensitive enough to find the virus that was there. And this story is very analogous to the case of the Boston patients, same situation where the virus came back from somewhere, but it was not readily detectable by the methods that we used. So we desperately need more sensitive tools to find every last trace of virus that exists in these individuals. So how are we doing that? So this is one of many approaches that we're utilizing. I think the simplest way to approach this metaphor is we will find more needles by sampling much more of the haystack than conventional methods. So we're utilizing droplet digital technology that enables analysis of 10-fold more cells and DNA than standard approaches. And this range of tools is something that we're developing in collaboration with Raindance Technologies based in Massachusetts. So I'm going to give you an example of some of the experiments that we've been conducting. So how can we test the sensitivity and specificity of these methods? One way to do it is to engineer these artificial cocktails in the laboratory where we can start with primary cells. So we actually take cells from an individual, from an uninfected individual that contain absolutely no HIV DNA. Then we have these special cells in the laboratory that each harbor exactly one copy of HIV DNA. So we can spike in these HIV-infected cells into a background of uninfected cells in known proportions, and we can see how well our methods do in the laboratory at pinpointing those fractions. So the first example I'll show you is starting off with just 1 million uninfected cells. So there's no HIV DNA in this picture whatsoever. And I know probably a lot of you are not familiar with reading this type of plot, but I'll explain it to you. I think it's pretty simple. Each one of these little pixels or points that you see right there is an individual droplet, okay? And those droplets are in one of 4 character states. In the bottom left corner, these droplets contain absolutely no DNA. In the top left corner, these droplets contain human DNA. In the bottom right corner, each one of these droplets contains HIV DNA. And lastly, sometimes you'll actually get more than one piece of DNA in a single droplet. So some of these droplets will contain both. So as you can see in this first example, there's absolutely no HIV DNA present, which is good because we didn't put any into the sample. So we know we're not getting any false positives. Skipping forward, now we're going to flood the system with HIV. And we have 1,000 HIV-infected cells that each harbor one copy of HIV. So sure enough, you can see both of these corners are lighting up. Because we have lots of droplets that contain HIV DNA and both human genomic DNA and HIV, as expected. This is the tricky one, and this is, I think, one of the most impressive things that have come out of this collaboration so far, is starting with over 1 million uninfected cells and spiking in a single HIV-infected cell. You probably can't see this one dot. But we've actually been able to capture and detect a single HIV-infected cell, or technically an HIV-containing droplet, out of this vast sea of millions of droplets that contain human DNA. So again, is this a perfect solution? No, but I think this is a very significant step in the direction where we'll be able to sensitively sniff out virus in individuals on suppressive therapy. Okay, so this might almost seem like a little bit of a non sequitur, but I'm an evolutionary biologist at heart, so I had to include this in here. But I think we all know that really one of the keys to HIV's success is that HIV has an extremely high mutation rate and really almost has the capacity to evolve around everything that we throw against it. So, a classic case would be evolution of immune escape. Another classic case would be with the early antiretroviral drugs. The virus could very, very easily evolve resistance to those drugs. And so here's the consequence of that. When we look at worldwide HIV diversity, this tree, the size of the tree on that slide, represents worldwide circulating HIV diversity. Now, that tiny little blob I'm showing you there is worldwide influenza diversity. And the worldwide influenza diversity is still a challenge in terms of clinical management of that infection. So this kind of gives you an idea of how ridiculously diverse HIV is. So why am I presenting this to you? Well, I'm going to tell you that there's actually a huge trade-off associated with the evolutionary prowess of HIV. So even though HIV relies on its evolutionary wizardry to be able to slither around antiretroviral drugs and the immune system, the trade-off is that the vast majority of HIV genomes in a human body contain mutations that render it inactive. So that's the cost. It has to have a very sloppy process at making copies of itself. So over 90% of viral genomes in an individual do not have the capacity to replicate or grow. The reason why that's relevant is that if we just use a simple counting approach to just count all of the viruses that are there, we're going to dramatically overestimate the size of the latent reservoir because the vast majority of that virus is not going to grow out when we stop antiretroviral therapy. So a key challenge associated with this field is to develop sophisticated methods to be able to accurately differentiate between replication-competent virus that can grow and replication-incompetent virus that's just a dud. So there's several different approaches that we're utilizing within this module to be able to do just that. I'll go very quickly over one of them. So as I mentioned, those viruses are defective because the genetic sequence of that virus has critical mutations that prevent it from producing proteins and prevent it from replicating. So we're going straight to the viral sequence. We're actually taking the viral genomes. We're using this Raindrop platform developed by that company, Raindance Technologies in Massachusetts that I mentioned. What we're doing is we're doing single molecule next generation sequence of all of the viral genomes that are in there. And then we're using a suite of bioinformatic approaches to predict whether each one of those genomes has the capacity of replicating or not. So what that's going to tell us is the overall proportion of viral genomes in a sample that has the capacity to replicate. Then we're going to use a quantitative approach that will give us an absolute count of all of the viral genomes in a sample. So if we put those two pieces of information together, this approach, this combination of droplet digital PCR and next-generation sequencing, will give us an absolute count of replication-competent HIV in a sample. So here's another approach I'd like to talk about that is being developed by my esteemed colleague and buddy, Dr. Tim Henrich. This is one of several different approaches that his group is developing to measure replication-competent virus. So here, instead of looking at the sequence and inferring the virus's capacity to replicate, he's actually growing the virus. He's taking infected cells out of a patient and culturing them in vitro to actually watch the virus grow. So that way he'll be able to measure the frequency of infectious virus in a clinical sample. Other people have similar methods, but the critical advantage associated with the method that Tim is developing, you'll notice here this nickel. So Tim has figured out a way to do this on the microscale, in miniature, on these little microfluidic chips, which really is a game changer because the conventional macroscale version of this is extremely labor intensive and extremely costly. But this approach that Tim is developing to grow virus in vitro should really allow us to cost effectively and rapidly evaluate the size of the latent reservoir using this approach. So another critical challenge is that we typically characterize latently infected cells in a vast sea of uninfected cells. And a huge challenge to this field for all four of our modules is we really need to understand the basic biology of the latently infected cell as it exists within the infected individual. But most of the time, we're just sort of measuring it in this amorphous sea of uninfected cells. So if we could develop methods to specifically zero in on these latently infected cells in isolation, I think it would teach us volumes about their biology, and it would also give us better tools to identify these infected cells within a mixed population. So we've had the distinct privilege of teaming up with Dr. Adam Abate, who is a professor here at UCSF in bioengineering, and he's designed this revolutionary single-cell technology called PACS, which stands for PCR-activated cell sorting. So I don't know how well you guys can see it in the back of the room, but what's happening right here, these little tiny dots are actually single cells. And each one of these single cells is being suspended in a single microfluidic droplet as they pass through this trench. So all of these droplets here each contain one single cell. So what can we do with those cells? We can actually perform PCR within each one of those droplets to look for particular nucleic acid targets, and then we can sort the droplets based on the presence or absence of that desired target. So in our case, we will be amplifying HIV DNA or RNA in each one of those cells, each one of those droplets, which again contains a single cell, and we'll actually be able to sort the population of cells from a patient into cells that contain the virus versus cells that don't contain the virus at all. So I think this will be a game changer in really allowing us to perform a very detailed characterization of latently infected cells in the near future. So another very key challenge, and Dr. McEwen just went through this in great detail, is that Most of the time in the clinic we rely on peripheral blood because it's very easy to access peripheral blood. But perhaps, unfortunately, I think the most critical sites associated with HIV persistence are in tissues rather than in circulating peripheral blood. So we desperately need methods to interrogate the virus within the tissues rather than peripheral blood. There's some obvious challenges, though. You can't just go in and harvest lymph nodes and harvest the spleen and the gut-associated lymphoid tissue on a regular basis. So we need to find proxies for the persistence in tissues. So here's one approach that we're investigating. Circulating antibodies in the blood may inform us about HIV persistence in tissues. So if we just think about the basic biology of antibodies, antibodies are continually circulating throughout our body, and they get to see or get to touch or detect virus in these different sites all over the body. So by studying the circulating antibodies in the periphery, they might actually yield information about the nature of viral persistence in these different tissue sites. So here's just a couple of quick examples of those data. One is, if we look here, this is kind of the natural cadence of the anti-HIV antibody response in infected individuals. So once you get infected, you have this massive increase in the titer of circulating antibodies against HIV. But when patients go on antiretroviral therapy, you can see that there's this very consistent decay in the circulating level of antibodies against HIV. But the antibodies don't disappear off of the map entirely, it seems. So one of the questions that we're entertaining is the level of anti-HIV antibodies in circulating plasma during antiretroviral therapy, does that level actually tell us something about how much virus remains in the body? And this is a very active area of investigation in our group. So another way of presenting these antibody data to you. So we're all very familiar with the case of the Berlin patient, which may be the only example of a true viral eradication, although it's hard to prove the absence of something. But if we look at the level of circulating antibodies against the HIV envelope protein in HIV-negative individuals, as well as several different HIV-infected populations, you can see that the level of antibodies targeting HIV in the Berlin patient is intermediate between uninfected individuals and infected individuals. So this raises the possibility that the antibody response is seeing something that other methods, that nucleic acid-based measures fail to see, which I think further reinforces this concept that the antibodies could be leveraged to tell us something about viral persistence in the setting of antiretroviral suppression. And just kind of to close with this technology, as Mike also mentioned and Rowena mentioned, I think one of the most exciting things that we have cooking in this module is the idea of using an imaging approach to directly visualize the virus in tissues in vivo. And this is how this approach would work in a nutshell. So this is a single HIV particle. The HIV virion or particle has all of these surface glycoproteins embedded in the surface of the virion, called the viral envelope, or sometimes also called GP120. So we can take antibodies that target the surface glycoproteins that are embedded in the virion surface, and then we can attach a radio label to those antibodies. Then we can infuse these radio-labeled antibodies against HIV envelope into individuals, and then we can detect the presence, we can detect the frequency and presence and distribution of these radio-labeled antibodies inside an infected individual using either immuno-PET CT or also MRI imaging. And so on the screen here, you can see an example of that, where these different colors pertain to the relative degree of persistence of the virus inside the individual. So Rowena also mentioned that a group at Emory has recently performed a very similar experiment in the macaque model. And so here you can see, using this approach, in the absence of antiretroviral therapy in the animal, you can see these large yellow patches which reflect areas of viral persistence. And then when you put the animal on suppressive antiretroviral therapy, you can see most of these yellow patches disappear off the map, but not all of them, suggesting that these methods are likely sensitive enough to detect the little bits and pieces of virus that persist during antiretroviral therapy. So to close with this, I think we're using a large variety of tools here that all have a decent amount of promise, but I don't think we'll really know if any of these tools are telling us anything that we need to know without seeing if they have the predictive capacities that we need within a clinical context. So ultimately what we want to be able to do is this. We want to measure something. We want to measure the latent reservoir using one or several of these tools right before antiretroviral therapy. And we want to see if the measurements that we take will allow us to determine whether it makes sense to stop antiretroviral therapy and whether the patient is likely to experience a very extended remission period in the absence of antiretroviral therapy. So to facilitate this, our whole group has patched into several different ongoing clinical studies that will involve an analytical treatment interruption, so we can actually do exactly that. We can use our tools to measure a range of values or parameters before the cessation of antiretroviral therapy, and we can see if those tools allow us to predict whether a cure is likely, or even predict the exact magnitude and nature of the ART-free remission period that we're likely to achieve.